We have made enormous advances in the last 20 years in the treatment of cancer, and this is largely due to the development of targeted therapies and immunotherapy. Over the last several decades, the DNA of tumors has been analyzed and critical observations have been made. We have identified mutations in genes that result in the uncontrolled growth of tumor cells. In other cases, additional copies of one gene or several genes has been identified, and this is referred to as an amplification. These mutations and amplifications lead to uncontrolled growth. And now we have medications that target these changes. For example, in breast cancer, there's amplification of a growth factor called HER2, and a medication called Herceptin has been developed to inhibit this factor. Herceptin is an antibody, a protein, that finds and helps to destroy cells that overexpress this factor HER2. This medication has saved countless lives, and this really is just one example. There are now targeted therapies in lung, liver, thyroid cancer, leukemia, lymphoma, and numerous other tumor types. And there are new and exciting therapies being developed. In addition, immunotherapies have made their way into the clinic. This is a way of activating the patient's own immune system so that it recognizes the tumor and eliminates it. These therapies are innovative and rapidly improving, providing great hope for the future. But while all these therapies have been revolutionary, a dilemma remains. The majority of patients do not have a targetable mutation or an amplification, and the majority of patients that are currently treated with immunotherapies do not respond. So how do we treat these patients? How do we develop new therapies to target their tumors? We have conventional chemotherapies, and these work by preventing cells from dividing. And this has been extremely successful when tumors are localized. But once a tumor metastasizes, once it travels to a distant site from where it started, we know that conventional chemotherapies almost never result in a cure. And it's important to note that nearly all cancer deaths are due to metastasis. We know that from clinical studies, as well as laboratory data, that metastatic cells are very different from primary tumor cells. They are more aggressive and they're more resistant to treatment. So what now? How do we treat patients with metastatic disease who don't have a targetable mutation and who aren't responding to immunotherapy? So I think that we need to better understand the differences between metastatic cells and primary tumor cells. We have to understand the characteristics that tumor cells can acquire to make them capable of metastatic growth. So in order to do this, we really need to better understand the process of metastasis itself. And it's interesting because metastasis is actually extremely inefficient. So tumors can shed thousands to millions of cells into the bloodstream each day. And nearly all of those cells die and are cleared from the circulation. In order to better understand metastasis, we need to understand those cells that are able to actually survive and grow at a distant site. And this is just a few cells. They can travel to the lung, the liver, the bones, and establish growth there. And we need to understand how they're capable of doing this when millions of other cells from the same tumor could not. So my studies are focusing on those metastatic cells. How are they different? Do they express different genes? While they may not have specific mutations, maybe they have a difference in the genes that they turn on compared to the primary tumors. In order to evaluate this, we can use RNA sequencing. But even though a gene is actually expressed, does not mean that the protein itself is active and important in the cell. So we need a way of determining which pathways are active and which pathways that are regulated by these genes are active. So in order to do this, we'll use mass cytometry. So this is a method that allows us to evaluate at the single cell level which pathways are turned on and which are not. By combining these two techniques, RNA sequencing and mass cytometry, we can start to identify new therapies in metastatic disease. There's a clear need for additional therapies to treat our patients with metastatic disease. Right now, our options are limited because our understanding of the biology is very limited. So I think if we can understand better the characteristics that cells can acquire, then we can target these, and we can help to turn our knowledge into a cure for our patients.